In this video, we will discuss the development of resistance by microorganisms to antimicrobial drugs, the mechanisms utilized to achieve resistance, and ways in which the development of resistance can be averted. As we have seen this semester, antibiotic resistance is a major problem in today's healthcare. When a population of microbes is exposed to an antimicrobial, some of those microorganisms will naturally be resistant to the drug. Resistance to an antimicrobial agent may be acquired due to random mutations that occur in the genes, or may be acquired on an R plasmid that has been shared by transformation, transduction, or conjugation. The image shown shows how a population moves toward one that contains primarily resistant microorganisms. Initially, the microbial population has microorganisms that are both sensitive and a few that are resistant. Under normal conditions, as represented on the plate to the far left, the sensitive microorganisms outnumber the resistant ones. This is because those that are resistant grow more slowly than those that are sensitive. The sensitive are therefore able to outcompete the resistant organisms for substrate and keep those that are resistant in check. When the environment changes and the antimicrobial drug is present, as in the second Petri plate, the sensitive microorganism dies while the resistant microorganism is not harmed by the drug. Please understand that the drug is not creating the resistance, but rather is selecting for the stronger resistant bacteria. This is an example of survival of the fittest, and in this case, the most fit is the resistant microorganism. If the antimicrobial drug remains present, the sensitive microorganisms are killed and are no longer available to compete with the resistant cells. In Petri plates 3 and 4, the population is now dominated by resistant cells that can no longer be controlled by the same antimicrobial. Microorganisms can resist antimicrobial drugs in a number of ways. Your textbook addresses 6 mechanisms of resistance. Microorganisms may resist the antimicrobial action of a drug by synthesizing an enzyme that acts to deactivate the drug. An example is the production of beta-lactamases. Beta-lactamases, also known as penicillinases, act to break down the beta-lactam component of penicillin drugs, making them inactive. The genes for producing beta-lactamases are often located on R plasmids that can easily be shared by horizontal gene transfer. Another mechanism employed by some microorganisms is to prevent the entry of the drug into the cell. Gram-negative bacteria are able to prevent the entry of many antimicrobials because of the presence of the outer membrane. The porin proteins of the outer membrane are responsible for preventing the entry of the antibiotics. Resistant cells may also alter the target of the drug so that the drug can no longer bind to it. Penicillins act by binding to a protein known as penicillin binding protein. Methicillin-resistant Staphylococcus aureus developed resistance by utilizing a modified penicillin binding protein that prevents the penicillin drugs from binding it to its necessary site so that it may act on cell walls. Some antimicrobial agents, as we have seen, act as antimetabolites and interfere with metabolic pathways. Resistance to such drugs is accomplished by altering the metabolic pathway. In some cases, they may even abandon the pathway. Some microorganisms have resisted the sulfonamides by abandoning the synthesis of folic acid and instead meet the needs by beginning to absorb it from their environment. An interesting means utilized by some microorganisms to resist antimicrobial drugs is the use of resistance pumps. Resistance pumps are proteins embedded in the microorganism's cytoplasmic membrane that allow them to actively pump the drug out of the cell. In some cases, the pumps work on more than one type of drug, allowing them to be multi-drug resistant. A final mechanism covered in your reading is a special technique employed by Mycobacterium tuberculosis as a means of resisting fluoroquinolone drugs. Fluoroquinolone drugs act to inhibit cells by binding to DNA gyrase and preventing replication. Mycobacterium cells that are resistant can produce a protein known as MFPA protein that binds to DNA gyrase in the same location, thereby preventing the fluoroquinolone drugs from working. The drug likely slows down cell division, but slow growth is better than no growth. Microbes can be resistant to more than one drug. When a microorganism is resistant to 3 or more drugs, it is said to have multiple drug resistance. There are a number of hospital as well as community-acquired pathogens that have developed multiple drug resistance. The development of these organisms have been facilitated by the exchange of R plasmids via horizontal gene transfer. These drug-resistant organisms are selected for in hospital and nursing home settings due to the constant use of drugs that eliminate the sensitive cells. Multi-drug resistant pathogens have also been referred to as superbugs because of the difficulty in treating them. In some cases, when a microbe develops resistance to one drug, it becomes resistant to similar drugs. This is known as cross-resistance. Your textbook addresses 4 ways that we can retard the development of antibiotic-resistant pathogens. The first is to utilize sufficiently high concentrations of the drug and maintain those concentrations long enough that all sensitive cells are killed and the resistant cells are inhibited long enough for the body's immune system to kill them. Again, remember, antimicrobial drugs are only used to inhibit the microorganism so that the immune system has an opportunity to kill the pathogen. This is the importance of taking the medications in the right dosages for the right period of time. Another way that the development of resistance may be retarded is to administer the drugs in combination so that microorganisms that are resistant to one drug might be killed by another. It is important to recognize that the drug should act in a synergistic fashion and not antagonistically. Synergism means that one drug will enhance the activity of another. An example of drugs that are used synergistically is the use of penicillin, which, as we've seen, disrupts the synthesis of cell walls, given in combination with clavulanic acid. Clavulanic acid is a chemical that acts to deactivate beta-lactamases produced by some resistant strains, thereby allowing the penicillin drug to work more effectively because it's no longer being degraded. If one were to administer drugs that work antagonistically to one another, you would reduce their effectiveness. An example would be the administration of a bacteriostatic drug with a cell wall targeting drug. Cell wall targeting drugs require actively growing cells in order to interfere with cell wall synthesis. The bacteriostatic drug would slow the growth of the bacteria, making the cell wall targeting drug less effective. A third way to retard the development of resistance is to limit unnecessary use of antimicrobial drugs. We have observed throughout the semester in some of your reading and your video assignments that we as a population have misused and overused antimicrobial drugs. We have seen that drugs have been overprescribed, prescribed for viral infections when they are ineffective against viruses, misused by the patient, made unnecessarily available to people in many developing countries to diagnose and treat themselves, and employed in the use of agriculture. Each of these areas of misuse and overuse need to be addressed to aid in the current problem in regards to antimicrobial resistance. A fourth way in which resistance can be averted is to work toward the development of new antimicrobial drugs. In some cases, scientists can use existing antimicrobial drugs to create variations of the drug. The development of new variations creates a newer generation of the drug. We have seen such work with the cephalosporins, in which there are currently 4 generations of drugs. Scientists also continue to search for new antibiotics in diverse habitats, such as the soil, marine microorganisms, and in or on other living organisms. A newer avenue for the possible development of drugs is to design drugs that are complementary to the active site of enzymes that would allow us to block the metabolism of pathogens without affecting human metabolism. This type of research is more easily accomplished because of the many advances that have been made in the areas of biochemistry and molecular biology.